Dear students, for class 12 zoology, this is biotechnology chapter. We are dealing with the process of recombinant DNA technology. The process of recombinant DNA technology or genetic engineering involves a large number of steps. The step number 5 first is how to isolate genetic material. Step number 2, amplification of gene of interest using PCR. Step number 3, preparation of the gene, how to cut DNA at specific sites where gene of interest is present. Step number 4, detection of the cloned gene, recombinant. Step number 5, how to obtain the foreign gene product. Step number 6, downstreaming process. So far the first step is concerned how to isolate the genetic material. Following steps are required for the isolation of genetic material DNA in pure form. Bacterial cells or plant cells or animal cells are treated with enzymes like lysozyme for the bacteria, cellulose for the plant cells, and chitinase for fungus. This will break the cell envelope open and macromolecules like DNA, RNA, proteins, polysaccharides, and lipids will be released. As in eukaryotic cells, the DNA is intertwined with protein molecules like histones. Additional proteins can be removed by treating with enzyme protease. RNA can be removed by treating with enzyme ribonuclease. DNA sample can be further purified by using additional extraction techniques. By addition of chilled ethanol, DNA precipitates out and can be observed in the form of fine threads in suspension. Agarose gel electrophoresis is employed to check the progression of restriction enzyme digestion. DNA being negatively charged moves towards the anode, positive electrode. Same technique is used for vector DNA. The second step involved is amplification of gene of interest using PCR, what we call as the polymerase chain reaction. Polymerase chain reaction technique was developed by Gary Mullins in 1985. If one knows the sequence, at least part of a DNA segment can be cloned. A number of copies of that DNA segment can be hugely amplified using polymerase chain reaction. It is able to generate microgram quantities of DNA copies, up to billion copies of desired DNA segment, present even as a single copy within the short possible time. The technique is based on the principle that when a DNA molecule is subject to high temperature due to denaturation, the two DNA strands separate. As a result, two single-stranded DNA molecules appear. DNA polymerase can copy these single-stranded molecules. This leads to formation of original DNA double-stranded molecule. Due to repetition of this process, several copies of DNA sequences can be formed. There are some steps involved in polymerase chain reaction. First step, DNA template desired segment to be amplified. Second, two oligonucleotide primers specific complementary to the sequence present at 3' ends of the desired DNA segment. These primers are oriented with their ends facing each other, permitting formation of DNA between them. Third step, high temperature, 790 degrees centigrade stable DNA polymerase. It is needed for the formation of new DNA. Usually used DNA polymerase for PCR reactions is Taq polymerase. These steps can be seen from this diagram where we follow the different steps. The first step is denaturation at 94 degrees centigrade. Second step is annealing of the primers. Third step is DNA polymerase, Taq polymerase. And the fourth step is extension at 72 degrees centigrade. And fifth step is amplification via the DNA of interest is obtained. The third step involves preparation of the gene, how to cut DNA at specific locations where the gene of interest is located. Gene cloning in bacteria is achieved by cleaving the purified DNA with enzyme restriction endonuclease, which produces small fragments, approximately 4 kilobase pairs. Each fragment has a sticky complementary single-stranded end. Eukaryotic genes contain introns that are not processed in bacteria. Therefore, DNA for cloning is usually obtained as a reverse transcriptase generated copy of DNA of the relevant messenger RNA. The third step involved is detection of the cloned gene recombinant. Cells with recombinant DNA are selected on the expression or non-expression of some traits like resistance to antibiotic chloramphenicol. Direct selection of recombinants is made due to encoding of these traits by vector or cloned DNA sequence. One more step is there. How to obtain the foreign DNA, foreign gene product. When alien DNA is inserted into cloning vector and then transferred to host cell, DNA gets multiplied. Due to expression of this foreign gene, proteins can be formed. Lastly, the downstreaming process. Products formed are separated and purified. Steps are collectively called as downstreaming processes. Suitable preservatives are used for medicinal purposes. Clinical trials are carried out. Quality control is also maintained. This is how do we process in the recombinant DNA technology or genetic engineering. That's all for today.